السلام عليكم ورحمة الله وبركاته. أنا أسماء المطيري من دفعة 222 واليوم حتكلم عن محاضرة Nerve and Muscle Physiology للدكتور عارف. حنبدأ بهالصورة واللي توضح أن ال interior of the cell is actually negative. So when you insert an electrode, it's gonna show that the interior of the cell is negative. Regardless of what cell it is, the interior of the cell would be negative and the outside would be positive. So the membrane potential is actually defined as the difference of the electrical charges between the inside and the outside of the cell. For the resting membrane potential, it really depends on the size of the nerve fiber itself. So large nerve fibers are gonna have around minus 90 millivolts. However, smaller fibers are gonna have less, which is around minus 70. So I will explain how the action potential actually works through this figure. So follow the dark black line and follow the steps with me right here. So first thing is that the resting membrane potential for the nerve fiber is around minus 70 for the small nerve fibers. The threshold for the nerve fiber is going to be minus 50. The second step, which is the depolarization, is gonna kick in when the sodium enters. So we need to open the voltage gated sodium channels through giving it an impulse. So the sodium enters and but the potassium is still closed. So when we want to move to the third step, we need to actually to open the potassium channels. For the potassium channels to open, first we need to reach around minus or plus 30 as a membrane potential. Then we need to close the Na channels. So the Na channel is going to open with the activating gate. However, it's going to close by the inactivation gate. So the third step starts, which is the hyperpolarization. So the sodium would go out of the cell. However, the sodium is not entering anymore. So the hyperpolarization ends and the action potential ends as well. But we want to return the nerve fiber towards the resting membrane potential, which is the normal membrane potential for the nerve fibers. So it's going to happen in two ways. First, the Na sodium or potassium channels pumps are going to pump 3 sodium out and make 2 potassium enters in. Another thing that participate in the process of returning the membrane potential towards normal, which is the resting membrane potential, is going to be when there's a switch between the activation gate and the inactivation gate. A little bit of positive ions are going to enter, which going to help in returning the resting membrane, the membrane potential toward the normal, which is the resting membrane potential. So moving on to myelin and myelinated nerves. So what helps in synthesizing myelin is actually Schwann cells. Helps in insulating and making the transduction or transforming the signals through the nerves much faster. Schwann cells would warp around the nerve when it's fully myelinated. However, it's going to partially warp and myelinated nerves. So as you see in this picture, you would see that in myelinated nerves, Schwann cells are going to warp around it completely. However, in unmyelinated nerves, it's going to just warp partially, as you see here. So now we're going to talk about the muscle physiology. We're going to look at the organization and the structure of the skeletal muscles. So the skeletal muscles are consisting of muscle fiber, and the muscle fiber are actually consisting of myofibrils. Myofibrils, when you take them and look at them under the microscope, you would see that it's consisting of building blocks, which is the sarcomere. The sarcomere is the building block between 2 Z lines. It consists of the thin actin and the thick myosin. So as you see here, myosin would have 2 molecules wrapped around, creating a tail, and have 2 heads. The head have actin binding site for the actin molecule to bind, and myosin ATP sites to cleave the ATP. And here, when you look at the thin filaments of the actin, you would see it consists of the actin at first, and tropomyosin, which is the brown line, and the troponin as a third part of actin whole molecule. So now we're going to discuss the length tension diagram. So when you look at it, it basically discusses the idea that what is actually the optimal length of a sarcomere in order for it to give the optimal tension. So the letters here would demonstrate the length of the sarcomere, and here the percentages of the tension. We're gonna look at the letter A, which is when the muscle is fully contracted. So when the muscle is fully contracted, the length of the sarcomere would be around 1 and 1.5 as a length, and the tension here is around 90%, which is not the best. However, we need it to be better. So B and C, which is around 2 and 2.2 microns, is gonna have around 100% as a tension, as you see here, which is the optimum. When you look at the muscle or the sarcomere, it's like fully stretched, which is around 3.5. You're gonna see like the tension is the least of them. So in order for everything to work, we need ATP as energy resource. So like the most important 3 steps that need ATP as a workout, we need the first thing is myosin ATPase activity to actually start a power stroke. And the second is for the detachment of the actin, the myosin, we need ATP to detach. And finally, the active transport of calcium back into the sacs of sarcoplasmic reticulum during relaxation. An important thing to mention is when the ATP is, or the head of the myosin is not detaching from the actin, we would have a condition called rigor mortis, which usually happen during death. So how can we get ATP? There are like around 3 resources to supply the whole process. First of all, creatine phosphate. When we add it to ADP, we would take the phosphate group and add it to ADP, we would have ATP, which is sufficient to supply from 5 to 8 seconds during contraction. The second way is going to be glycolysis, which is the synthesis of ATP without an oxygen. The third step, which is the most important step, is oxidative metabolism, which is the use of oxygen to synthesize ATP. Finally, we're going to discuss the motor unit. So the motor unit is basically a single nerve, how many muscle fibers are supplied by a single nerve. So in a more simplified way, how many muscle fibers are united under the umbrella of a single nerve. As you see here, this picture, a single nerve have supplied around 3 muscle fibers. That's it for me. Hope it helps. Thank you for listening. Best of luck.